A very good evening to all the participants. This is the first week live practice session for the NPTEL course Environmental Biotechnology. I am Guru Priya, Prime Minister's Research Fellow in the Department of Civil Engineering, IIT Madras. I have my bachelor's and master's in biotechnology, and currently I'm working in the field of environmental biotechnology. So, my research focus is on anaerobic digestion, on enhancing the biomethane production through anaerobic digestion. This is a quick intro about me. So, with this, we'll start the session. So, in this live sessions, we'll first have introduction for the week's lecture first, and second of all is the live session pattern. So, few sessions will go on like question and answer sessions, while few other sessions will be theoretical sessions. And if you have any doubts in between, you can either unmute and ask, or you can post it in the chat box. So coming to the week 1 course content for the environmental biotechnology course. So it has introduction to environmental biotechnology. It discusses the role of different microorganisms, and it tells about the global crisis that involves environmental challenges, and it highlights the need for environmental remediation. So, in order to go in detail about this environmental remediation, we need to know about what is an ecosystem, what is it made of, what are the different components of the ecosystem, and how the understanding of this ecosystem is going to guide us towards environmental remediation. So, this is the whole idea of the week 1 content. So, the first thing that we'll be seeing is we'll try to compare biotechnology and environmental biotechnology. So when we go about comparing biotechnology and environmental biotechnology, it relates biological sciences. So biotechnology relates biological science, engineering, and finally it yields a relevant application. So we will have a basic understanding of the sciences, the fundamental principles. We will try to extrapolate what are the engineering aspects of it, and by combining these knowledges, we will try to develop a relevant application in terms of a product or a process. When we talk about environmental biotechnology, we are going to use the principles of biotechnology in the environment and for the environment. So, since both deals with nature, the principles can most often overlap, where the principles can come from the environment, which is the nature. I hope you have heard about something called biomimicry, where many of the tech that we build today is kind of replicating the uh, biological systems, like bullet trains, velcros, all these. So the solution can be right in front of us. We need to identify it, we need to identify the principle behind it, and maybe we need a little amount of research and work to scale it or to bring it into application. So that is where we have this biotechnology and comparison between biotechnology and environmental biotechnology. So the next question is, how does environmental biotechnology contribute to sustainability? So environmental biotechnology, as we saw in the previous slide, is going to deal with biological systems which are present in nature. And sustainability, when we talk about some natural solutions and all. First of all, we'll see what is sustainability. Sustainability is meeting the needs of today without compromising the potential to meet the needs of tomorrow. So that is what we call it as a sustainable process or a sustainable product. So by use of a sustainable product or process, we'll be able to uh, fulfill the needs of the current generation, yeah, provided we are not going to exploit the use of the resource by the future generation. So sustainability is a very key aspect for uh, a healthy sustenance of humankind, of the environment, of the nature, and of the entire world. So sustainability involves three factors. One is bearability, equability, and viability. So when we talk about bearability, how much can we bear? In terms of environmental biotechnology, bearability is going to talk about how much of the load can this environment take, uh, above which we are going to harshly exploit the environment such that we are going to cause a irreversible damage to the environment. And when we talk about equitability, um, if you are going to work on environmental biotechnology, we should make sure that the access for it is equitable to everyone, irrespective of the social class, economic class, or irrespective of whether it is a human or animal. Everyone should have the right to enjoy the benefits of environmental biotechnology, and that is the concept of equitability. Viability is whenever we find a tech or a product, let's say, if it is going to be higher than the purchasing capacity, then the product or the tech is going to fail. If only if it is going to be affordable, it is going to reach people, and because we have some amount of consuming capacity, only up till that we'll be able to consume a process or a product. So in that case, building a tech which is of low cost, or a product which is of affordable cost maybe, is very important. So a nexus of all these three in equal proportions, which is bearability, equitability, and viability, is very important. So the three aspects that connects out of the three aspects that connect the sustainability, environment is one among them. The other two are social and economic aspects. Like if you talk about environment, we should check whether whatever we are going to develop is going to be bearable and viable. But when you come on discussing about this equitability, there is where you get this aspects of social and economic factors. So the next question is, what are the applications of biotechnology in environmental processes? So in environmental processes, we have the application of biological aspects, physical as well as chemical aspects. So especially pertaining to biotechnological aspects, what is the application in environmental processes that we can see? So one is waste treatment. So we have solid waste, wastewater, we even have polluted air. So all of this we can treat. So out of this waste, we call it as solid waste and wastewater mostly. So we will surely have biological treatment units in the solid as well as wastewater treatment systems. And in order to understand the environmental processes, this nutrient cycling is very important. So, right, starting from carbon, nitrogen, which are very major. Uh, we know about carbon cycling and nitrogen cycling in the environment. And all of this has microbial intervention in it. So, in order to understand the mechanism behind this, in order to understand the significance of this, We need the knowledge of biotechnology to understand such environmental processes. And next one is about the bioremediation. So the difference between waste treatment and bioremediation as such is, in waste treatment we are just talking about effluent of any process, like industrial waste, municipal waste, like that. In case of bioremediation, there may be some kind of pollution that is happening, or there might be the intervention of inorganic pollutants. So in waste treatment, you mostly go for complete treatment of organic pollutants, that is called mineralization. Whereas in case of bioremediation, you go for containment of the toxicity or containment of the pollution. For example, if there is some metal based pollution in our water system, how do we remove the metal? We can't like eliminate the metal, or we can't destroy the metal. We can just separate or remove the metal from that particular system. And one more thing is bioleaching, which is biological extraction of metals from wastes as well as ores. So this comes under bioremediation. And the next application is fermentation. This is a broad term. So we use fermentation for production of various products, and energy. So products, as we all know, it is going to be um, alcohols, fatty acids, volatile acids, ketones, aldehydes, and many other chemicals. So in biorefinery terms, you call it as platform chemicals or product chemicals. Like platform chemicals, in the sense, these are intermediate chemicals which can be converted by different processes into a array of final usable chemicals, or you can use the platform chemical as such. The final end product chemical is again used in a multitude of purposes. Like the most important product is currently ethanol. uh, we see this EBP, ethanol blended petrol. So that is one of the main fermentation products. And next, talking about the energy. So how can we derive energy from fermentation? That is by anaerobic digestion. So majorly, where we try to get biogas, biohydrogen, biomethane, whatever. So biomethane, as we all know, has its own calorific value. It can act as a renewable energy source or a renewable fuel. Similarly, research on hydrogen is also accelerating. So these can act as energy source. And similarly, one more product is biodiesel, which we get by transesterification. So all these are fuel and energy related products. And this can also reduce our reliance on renewable fuels. And finally, we can do environmental monitoring with the use of biotechnology in environmental processes, as in we can develop biosensors, and we can have a regular monitoring of the system. And one more aspect that hasn't been discussed here is the knowledge of native microorganisms. So that will be extensively studied in all the lectures that are coming in the future. So we know that there is some microorganism driving a process which is environmentally beneficial, but how do we know what is the reason for that to happen? What is the mechanism behind that happening? What if the environmental condition is going to change? How the reaction is going to change? So all these factors are also uh, studied using biotechnology only. Uh, so it involves molecular studies, biochemical studies, such that we classify microorganisms based on their structure and functions. We group them, and we finally try to elucidate how a community functions, and we try to extrapolate this function to remediation processes. So all these are the applications of biotechnology in environmental processes. So next is, how is the scope of environmental biotechnology defined? So scope in the sense, environmental biotechnology as a term can incorporate everything in the world because everything deals with environment. But when we are going to conduct studies, we should be defining the scope so that we have a directed approach in understanding a process or developing a process. So that is why we will be defining the scope of environmental biotechnology. So as I mentioned, environmental biotechnology can be related to biochemical engineering, which deals with the processes, how the processes works, how to control the processes, how to regulate them. It deals with electronics, where electronics is used to study biological systems. Biological studies inspire development of electronics, or bio-based products are used in electronics. So again, it's a two-way relationship. And when you talk about food science, food technology, and all, obviously, agriculture, food, and environment and biotechnology are very highly interrelated because we deal with complete biological systems like microorganisms, plants, and animals in this. So, here comes the intervention of products, processes, techniques, and many other aspects. Instrumentation, all these comes into picture in this intersection. And next one is the material science. So material science, as I told, we have multiple biological materials or bio-based materials derived from biological systems that are highly being researched on. For example, biodegradable wrappers, bioplastics, so which reduces the load on the pollution or waste management in the environment. So when you see in the top portion, you have this microbiology, biochemistry, genetics. So these are the common areas which are used in understanding the system, understanding any biological system. For example, uh, when you talk about microbiology, it allows us to understand what microorganisms are present. When you talk about biochemistry, it allows us to understand how the microorganisms function, what are the products they produce, what is the chain of events that happen that leads to the production of a product. And when you talk about genetics, it gives us an idea about how evolution happens. For example, evolution in microorganism is very very rapid compared to macroorganisms. So when you know the genetics, you can easily predict what is the metabolic capability, how will this change if the environment conditions are going to change. So all this understanding you get it through genetic analysis. And finally, when you talk about computer sciences is very important because there is a concept of high throughput analysis. So when you talk about microorganisms are in millions and millions of numbers. They are not just one or two. So you need robust techniques to simultaneously study all of these microorganisms, right? So that is where your computer knowledge comes into picture. So you have this informatics, data analysis, so where you're able to get a large amount of data, process it, and draw conclusions out of it. So that is one aspect where computer science is involved. Another one is modeling and prediction of processes, monitoring of processes. In all these fields, you have the intervention of computer sciences. So this is how we define the scope of biotechnology. When we talk about the different, so this is a representation of the different fields in biotechnology, out of which environmental biotechnology is one, and it is highly interrelated with all other fields that are mentioned here. And if you observe closely, each of the field mentioned here is greatly related to any other field that is mentioned here. For example, when you talk about industrial biotechnology and nutritional biotechnology. So, industrial biotechnology largely focuses on developing processes, monitoring them, developing reactors, uh, defining process conditions, getting a stable process running. All of this is what we ensure in industrial biotechnology. When you talk about nutritional biotechnology, if you are going to produce some kind of nutrients, let's say some vitamins, so that's where you are going to have a connect between industrial biotechnology and nutritional biotechnology. So you have to remember that whenever you talk about any sector in biotechnology, you can't stand alone talk about it. You will indeed have a interrelationship between all these fields. So in order to define the scope of environmental biotechnology, it's very, very vast. Anything that happens in nature can be studied under environmental biotechnology, right from diseases to remediation to space, everything. So next one, we'll be discussing about the roles and challenges in environmental biotechnology, the current challenges in environmental biotechnology. So the role of environmental biotechnology currently majorly focuses on pollution prevention and abatement. Like, we know there is a large amount of pollution happening. How are we going to treat the pollution, and how are we going to avoid the pollution? We directly cannot stop polluting, but it's high time that we greatly decelerate the rate at which we are polluting the environment. So the major role is on pollution abatement and prevention, and next comes the remediation, which includes waste treatment and all. So yes, we have to prevent pollution, we have to find strategies for remediating, and finally we need to focus on resource generation and management. So there are some resources that are currently present. We are exploiting many resources. We are maybe we should judiciously use all these resources so that they are sustainable. Uh, if not, we have to find ways in which we can have medium for resource generation or resource replacement. And in the meantime, we can also find out sustainable ways to manage these resources so that we also decelerate the rate at which we are using these resources. And so, based on this, the challenges that are currently hindering the establishment of environmental biotechnological techniques is the population size and distribution. So, in the entire world, we have a large amount of population, but the distribution is very random and uneven. So, and also the societal, economical, and cultural background of people is also highly diverse. So, whenever we develop environmental biotechnological environmental biotechnology or technology basically, we need to check whether it adheres to the beliefs of the population. So in that case, mostly we need tailor-made or specific solutions. And when we are going to apply it to some other place, it should be modular such that we can modify it based on the needs of the second place in which we are going to apply the same. So that is one of the greatest challenges. Second is the rate of climate change. So the effects of climate change is highly unpredictable now. Uh, so how to understand this, how to go about this is a great challenge. And last one is the economy, as like population economy is also uneven. So economic support for the development of environmental biotechnological solutions is one of the greatest challenges. Okay. So, with that general introduction on what is environmental biotechnology, we'll now see the significance of ecology. So, this is where the content starts. So, ecology as such means study of life at home. So, which means home is the habitat. You're going to study about someone in their natural habitat, in their home. So, that is ecology. So, you see a mountain, you see the peak of the mountain, you go there and you take samples and analyze the microorganisms in that place. So there will be some native microorganisms that are present in that peak of that mountain. You go deep into ocean, collect some samples and check the microorganisms. There might be another set of microorganisms that are present in the deep marine environment. Ecology is the study of life at such habitats. Okay. It To be very specific, life includes all biological systems and not only microorganisms. So this includes microorganisms, plants, animals, all of this. And it does not only confine to microorganisms. So ecology is the study of life at home. Ecosystem, on the other hand, so when you talk about a system, it's going to be some framework. So ecosystem is going to provide us a framework to understand how energy and material flows in this environment. So when you imagine a colony home, let's or an apartment maybe, different people coexist there. You have neighbors, you have people on the top floor, you have people in the bottom floor, you have the security guard, you have a plumber, you have a secretary of the apartment. So you have multiple people in multiple roles. So, likewise, in any natural environment, different microorganisms are present and they are going to interact with each other. And there is going to be a set of observations which will reveal how energy and material is going to flow in the environment. Obviously, it is not going to be very linear, it's going to be a highly webbed matrix. So ecosystem is going to provide us the framework to understand this matrix of energy and material flow. So one important aspect about ecosystem is it not only tells us the interaction between living organisms, like how I interact with my neighbor, how I interact with a tree, how I interact with a dog in the apartment. No, only not only that, it also tells how I interact with the abiotic components. For example, how I interact with um, the temperature, how I affect temperature, how temperature affects me. So all these interrelationships also comes under the ecosystem. So this is the definition about ecology and ecosystem. So next we'll talk about how are the components of ecosystem defined. One is the trophic structure. So as you all can see here, trophic structure has two classifications. One is autotrophic and heterotrophic. So it's obvious that autotrophic organisms can synthesize their own food and heterotrophic organisms cannot. So the basic reason is autotrophic organisms have mechanisms through which it can assimilate inorganic carbon sources and then convert them to organic sources. Whereas in heterotrophs, this biosynthesis of organic compounds is not highly possible. So it directly feeds on organic substances that are present in nature. So the next one is structural level, where you have biotic and abiotic components. So biotic components are living components or biological components. Abiotic components are non-living. The chemicals, temperature, pH, etc. And next we have the functional components. The functional components mostly deal with the activity of the biotic components. For example, the energy flow, material flow, spatio-temporal flow. So spatio-temporal flow means spatio-temporal, if you split it, it is space and time. So imagine a forest. You take a sample from one end of the forest, in the middle of the forest, and in the farther end of the forest. It's not going to have the same composition of microbes. So there is going to be spatial variation depending on various factors. Similarly, if you collect sample today, a week later, a month later, and a year later, it's not going to be replicable. So that is the temporal variation. So with space and time, how things vary. That is spatio-temporal flow. And next comes the nutrient flow. So there is a key difference between material flow and nutrient flow. Material can be any chemical. Basically, if you talk about nutrients, it's going to be building blocks like carbon, nitrogen, phosphorus, potassium, sulfur. All this we will be taking it as a nutrient flow. And finally, you have the evolutionary flow. So this is to some extent related to the spatio-temporal flow, where based on the changing environmental conditions, how the microorganism is trying to evolve and trying to fit into the system. So this is the overall chart which can summarize it. In abiotic components, you have climatic and edaphic components. Climatic components include rain, sunlight or light, wind, and temperature. In edaphic components, you have soil, pH, minerals, and topography. When you talk about biotic components, when you talk about biotic components in an ecosystem, automatically three words come into our mind, which is the producers, consumers, and decomposers. We have different classifications like primary, secondary, tertiary, and quaternary consumers. The producers are the autotrophs which can take up inorganic carbon and synthesize organic materials, and this consumers are heterotrophs which rely on the products produced by producers, and once when these are dead, decomposers are the organisms that feed on this dead matter and ensure there is proper cycling and decomposition. So these are the different components of ecosystem. Okay. So yeah, I think in previous 2 to 3 slides we have talked about material and energy flow. So how do we differentiate material and energy flow in an ecosystem? It's very simple. Nutrient flow is cyclic, whereas energy flow is linear. So when you talk about a nitrogen cycle, the nitrogen is fixed as ammonia or nitrate. Again, it is denitrification happens and goes into the atmosphere as nitrogen. Nitrogen is again fixed and it goes back as nitrogen. This is a cyclic process. So usually nutrient flow is going to be cyclic, whereas energy flow is going to be linear. You are not going to get back stuff. It just keeps on passing from one component of the system to another. Now we'll see a few questions. So in a food chain, more available energy is observed at which level? So the answer for this question is at the beginning of the chain. So when you talk about the producers, that is where you will see maximum energy. So in a food chain, more available energy is observed at beginning of the chain. And based on energy acquisition, how are food chains classified? So remember, the question is reading based on energy acquisition. So we have two types. One is the grazing food chain and another one is the detritus food chain. So grazing food chain is energy derived from plant biomass are the producers. So producer is going to generate food consumer is going to consume on it. This part of the chain is the grazing food chain. When you talk about the detritus food chain, once the producer or consumer is dead, it has to be decomposed, right? So there are organisms that feed on the obviously the assimilation in these biomass is also nutrients and energy only. So there are microorganisms which can uh, feed on these nutrients for their survival, and the remaining they release it to back into the environment itself for the next cycle to start and take place. So based on energy acquisition, we have two kinds of food chains. One is the grazing food chain, which starts from the plant biomass. Another one is the detritus food chain, which starts from the dead organic matter. And both of this should go hand in hand to have a stable environment. Stable ecosystem. So till now we have seen about ecology or ecosystem in general. Now we'll see what is microbial ecology. So when we talk about microbial ecology, we need to know about the occurrence of the microorganism. What are the different kinds of microorganisms that are present? Uh, Considering the total to be 100%, what is the proportion of each of the organism that is present? How it is distributed in terms of space and time? So first of all, we have to study what are the microorganisms that are present. So that is the occurrence study. And next is the structure. So once we know what individual organisms are present, we try to elucidate what is the structure of that ecosystem. So for this, you also need the help of the knowledge on the community function. So you need to know what is the role of each of the microorganism, how one role can correlate, a role of one microorganism can correlate with the role of another microorganism. This will help you build a matrix or a map, just like a food chain or a food web. Hence, first you are studying about what is the microorganism present, what is its function, and then you try to derive a correlation among these organisms. And finally, you see what are the effects of the existence of microorganisms in such fashion. And when you know this, it becomes easy to predict if you're going to change the environment in certain manner. Let's say I have an environment that is at pH 7, it works in a certain way. If I'm going to drop the pH of that environment to pH 3, I will be able to predict how the response would be. Or if I'm going to spike the pH to pH 9, I should be able to predict how it will work. So this knowledge will help me develop tailored processes for remediation. Let's say I have to treat some acid mine, or I have to treat some organic waste, I have to treat some metal waste. So how should I go about choosing the proper microorganism? So for this, we need the knowledge on the occurrence, structure, and function of the microorganism, and this will constitute the microbial ecological studies. So the next question is, what is the need to study microbial ecology? So the need to study microbial ecology is that because it is ubiquitous. It is present everywhere, every time. And there is a lot of abundance and diversity in it. We have n number of microorganisms and different types of microorganisms. So we need to understand all of this. The complexity is very high. We need to understand the complex system, and that is why we need these studies. And next is the development and evolution. Next one is to understand the development and evolution. We need to understand how an ecosystem is being developed, or how it is becoming stable, what qualities it is expressing or acquiring in order to survive in the environment, what qualities it stops expressing or it inhibits in order to survive in a particular environment. All this understanding comes under development, and then you have the understanding of evolution over a period of many years, how this transition has been. So These are the needs for us to study microbial ecology. So the next question is, what are the possible biotic and abiotic interactions? So when we talk about interactions, first, when you just go in a zoom in zoom out manner, just imagine you have a cell and you're looking at a cell, okay, a single cell. When you're going to zoom in into the cell, you will have genes. So what is the interaction between the genes? So there is certainly interaction between the genes, or interaction of the genes with some proteins or with some communication signals from the environment that can turn on a gene or turn off a gene. So turn on, turn off, exactly what it means is the expression of the gene. So we all know uh, DNA gives us RNA, which further gets translated into protein. So when we have a gene alone, it's not necessary that that protein is going to be produced. So we have a DNA. If it is going to convert into mRNA, then maybe we can be sure, okay, that's going to be expressed as the protein. Not all genes are going to be expressed, right? So this is the interaction at the genetic level. And next you have this cellular level interaction, where again the signaling molecules act between different microorganisms. They help the microorganisms communicate. Microorganisms of the same species can communicate, of different species can communicate. So that is the cellular level interaction. And not only about microorganisms, and then you go about uh, population level interaction, where population is a group of microorganisms that are present in a habitat, a group of same species that are present in a habitat. So how they behave, that you term it as population. And a group of all organisms that are present, you call it as a community. And when you define the specific area for it, you call it a biofilm. The way it acts, you term it as a niche. And the entire place, you call it a habitat. And further, if you zoom out, it's going to be landscape. Different genes together form cells, cells together form population, populations together form a community, different communities together form a biofilm, different biofilms together form a niche, which in turn forms the habitat and then the landscape. So this is how there is going to be the biotic interactions, and for each of this, the major driving factor is going to be abiotic factors like temperature, pH, pressure, all of it. So next is about what are the possible microbial interactions. So we might have seen this earlier. First one is competition, inhibition, or cooperation. In terms of competition, it's about who's the fastest. There is a resource A, microorganism X can also feed on it, Y can also feed on it. But if X is faster than A, then the stability or the growth rate of X is going to be higher than Y. So this is called a competitive interaction. When you talk about inhibitory interaction, it might sound a little similar. The key difference is the presence of one is going to hinder the presence or growth of another microorganism. For example, we know the concept of antibiotics, right? So why do microorganisms inherently produce antibiotics? In order to uh, tamper the growth of any other microorganism, right? So that is known as inhibitory interaction. Third one is the cooperative interaction, where both have mutual understanding. One does not adversely affect the growth of another, or it may positively benefit the growth of another also. That is cooperation. So in this we have mutualism, commensalism, parasitism, and symbiosis. So when we talk about mutualism, it talks about mutual benefit for both the interacting organisms. So not only one is benefited, both of them are benefited only, so there is no negative anywhere. When you talk about commensalism, it is a interaction where one organism is benefited and other organism is neither benefited nor harmed. So that phenomenon you call it as commensalism. And then you have parasitism. So as we all know, one is going to affect the growth of other. So that is parasitism. An organism A grows at the cost of organism B, which means A is benefited whereas B is harmed. That is parasitism. And next, there is this term called symbiosis. So, symbiosis is just the interaction between two organisms. And in general terms, symbiosis refers to a mutually benefiting interaction. So, where it benefits both the organism. One more thing is syntrophic interaction, where it is similar to cooperation. For example, there are three microorganisms present. Microorganism A can convert uh, the substrate into a particular product, but it cannot completely mineralize it. Microorganism B can feed on the product of microorganism A and convert it into some other product, whereas microorganism C has the ability to completely mineralize the product of B. The C microorganism has the mineralization potential, but it cannot utilize the initial substrate itself. It needs the product of the second step. Only the product of the second step can be processed by the microorganism C. So in this case, there is coherence formed. Like first A acts on the substrate, then B acts, then C comes, and finally it get mineralized, and the needs of all three organisms are satisfied. So this interaction is known as syntropic interaction. So these are the different possible microbial interactions that can occur. So finally, we have seen about nature and environmental biotechnology. We have minute differences as well as a lot of similarities. We will discuss this in the upcoming lectures. Thank you for your patient listening and for joining in for the session. Thank you.